Hi everyone, this is Steven, and today we will be having a discussion about the introduction to medical microbiology and all its subfields. Let's begin with a review of your immune system, with focusing more on the body's responses to invading microorganisms. Also, in this lesson, in this core series, we will be covering bacteria, and then a series of lectures will be presenting the general concepts of your bacterial microbiology. And then there are sections also that we will be dealing with bacterial pathogens of humans. Similar sections such as your virology, mycology, and parasitology will also be covered. In each of the lecture series, there are introductory chapters that will stress the mechanisms of infection, characteristic of the type of your microorganism. Thus, it will provide you guys, the listener, with a framework of understanding rather than rote memorization of the clinical behavior of your pathogens. The final section of this lecture presentation series will be focusing more on the organ system and provides transition for clinical considerations. First, let's talk about viruses. As a part of medical microbiology, more than 2,000 species of viruses have been described, and with approximately 650 infecting humans and animals. Infection can lead either to rapid replication and destruction of your cell, or to a long-term chronic relationship with possible integration of the viral genetic information into your host's genome. The factors that determine which of these take place are only partially understood. In the next figure, you will see the clinical manifestations or overview of the clinical infections for viruses. We have in your nervous system, especially your CNS, okay, the encephalitis or meningitis, common cold such as your rhinoviruses, pharyngitis, gingiostomatitis, cardiovascular, especially for the Coxsackie B virus, hepatitis, A, B, C, D, and E, skin infections such as your smallpox, sexually transmitted diseases such as your herpes simplex type 2, human papillomavirus, and HIV, pancreatitis, your Coxsackie B virus, gastroenteritis, myelitis, pneumonia, and eye infections. What you're seeing in the background is a scientifically accurate model or atomic model of the external structure of your severe acute respiratory syndrome coronavirus 2, or the SARS-CoV-2, which is a strain of your coronavirus that caused the coronavirus disease, or what we call as your COVID-19. This was first identified in Wuhan, China during the December 2019, and each separate locus, or all of these amorphous blob, is an atom of cobalt for the membrane, crimson color for the E protein, green is for the M protein, orange color is for the glucose for the glycan, and turquoise is for the S spike for the glycoproteins. Viruses are one of the most smallest infectious particles, ranging in diameter from 18 to 600 nanometers, while most of the viruses are less than 200 nanometers, and they could not be seen using bright field microscopy or the light compound microscope. The genome of human viruses consists of either your deoxyribonucleic acid and the ribonucleic acid. The viral nucleic acids required for replication are enclosed in a protein shell with or without a lipid membrane envelope. So your viruses could either be envelope or non-envelope. Viruses are true parasites. They require host cells for replication, and the cells they infect and the host respond to the infection dictate the nature of the clinical manifestation. Viral diseases can range from benign, common cold to life-threatening Ebola, with acute, chronic, and even cancer-promoting presentations. Your immune response provides both protection and pathology and may be the primary cause of the illness. Viruses often initiated with non-specific flu-like symptoms caused by the host responses to the virus in the blood, and viral disease is characterized by the target tissues that is being infected by the virus. Showing you in the background is an HIV electron micrograph. Okay, for viruses, the classic symptomatology guides the diagnosis with the confirmation by isolation and cell cultures. It's really hard to culture, especially if the laboratory is not in BSL-4. Okay, detection of viral components or your antiviral immune responses with a prominent role in genetic detection and sequencing. Well, nowadays treatment for viruses has advanced, so there is now a tolerable cure for hepatitis C virus and a lifelong maintenance of your human immunodeficiency virus infections. We call it the antiretroviral drugs, or the ARV. There are also new vaccines, immunizations that have reduced the risk for several viruses, and vaccines for the human papillomavirus and hepatitis B that are also preventing your cancers. We will talk more of these viruses in the future lessons here in medical microbiology. Bacteria, okay, they are deceptively simple in structure. They are prokaryotic organisms, which are simple unicellular organisms with no nuclear membrane. So if you study on its cell biology, they don't have mitochondria, they don't have Golgi bodies, or your endoplasmic reticulum, and they reproduce by asexual division, specifically binary fission. Using this figure, you will see the overview of your bacterial infections, starting from bacterial meningitis and the genera and the species of your bacterium that causing the disease. And there are not just one, but a lot of bacterial species that causes bacterial meningitis, otitis media, pneumonia, skin infections, sexually transmitted diseases such as your urinary tract infections, food poisoning, gastritis, upper respiratory tract infection, sinusitis, and also eye infections. Most bacteria have either a gram-positive cell wall with a thick peptidoglycan layer or gram-negative cell wall with a thin peptidoglycan layer and an overlying outer membrane. So you can check that one using your one of the staining techniques that is being used in the laboratory, which is the gram stain technique. Bacteria such as your Mycobacterium tuberculosis have more complex cell walls, and some other bacterium lack this cell wall structure and compensate by surviving only inside the host cells or in a hypertonic environment. The cells 1 to 2 antimicrometer or larger shape, it could either be spheres, rods, or spirals, spirochetes, in spatial arrangement, including single cells, chains, or clusters, are being used as your preliminary classification of bacteria, and we solely based on the cultural characterization of the colonies that are found in your solid or semi-solid or liquid medium. Okay, and we also have phenotypic and genotypic properties of the bacteria that is forming the basis of your definitive classification. Genetic materials or analysis methods are becoming faster and cheaper, and the influences of specific types of microbe within the biome on our immune system, metabolism, drug metabolism, behavior, and general health are uncovered. Of course, we live in a microbial world with microbes in the air we breathe, the water we drink, the food we eat, and many of which are relatively virulent, but some of which are capable of producing it. Okay? The human body has inhabiting by thousands or different types of bacterial species, and they are living in a permanent parasitic relationship. These population of microbes residing in our intestines, on our skin, and other mucoepithelial surfaces is what we call as your human microbiome. And almost an entire organ, some scientists suggest that it could be another organ system in your body, your microbiome. And each of us harbor a unique microbiome. It's similar to a fingerprint, okay? All has similarities but individual differences. Although it's influenced by our genetics and policed by our immune system, the microbiome is somehow sensitive, okay? This is sensitive to the environment, to our diet, and the antibiotics and drugs we use. In the near future, we will see an increased use of therapeutic manipulation of the intestinal microbiome with fecal transplants beyond the current treatment of the recurrent Clostridium difficile colitis to correct your inflammatory bowel diseases, type 2 diabetes associated metabolic syndrome, and other diseases. Before we move on with the next slide, let's answer this question. Why is it necessary to correctly identify the germs causing the infections? What is its intent? You may pause the question and try to answer using your own stock knowledge. You're right. Bacterial diseases can result from the toxic effects of bacterial products, your exotoxins, or generally we call it toxins, or when the bacteria invade the normally sterile body tissues and fluids. Some bacteria are always pathogenic, expressing the virulence factors that causes tissue damage, and whereas other cause diseases by stimulating inflammation, and many do both. Okay, we need proper identification of the infecting bacteria, and it also allows us for predicting the disease course and the appropriate microbial therapy. Unfortunately, a lot of people are inappropriately using antimicrobials and other factors that have led to the selection of multiple antimicrobial resistant bacteria that cannot be treated. We move ahead with the fungi. The eukaryotic unicellular organisms. This is a micrograph of aspergillosis in H&E stain. You notice that Aspergillus hyphae may have a uniform thickness, and they typically branch at a 45-degree angle. And these are the study of the fungal infections is what we call as your mycosis, or mycosis in general is the fungal infections that infects humans. These are the following examples. We have oral candidiasis, we have tinea corporis, Theriasis vesicolor, onychomycosis, and eumycetenoma. While fungi, in contrast to bacteria, when it comes to their cellular structure, is quite complex. They are also referred to as eukaryotic organisms. They have a well-defined nucleus, they have mitochondria, Golgi bodies in the ER. They can exist in either unicellular form in yeasts, which can replicate asexually, or in a filamentous form, which we call as your molds, which can replicate asexually and sexually. Okay. Some fungi have a mold form in the environment and a spherical form in the body at 37 degrees Celsius. These are known as your dimorphic fungi and include such organisms as histoplasma, blastomyces, and coccidioides. In contrast to your bacteria also, fungal infections range from benign skin infections to your life-threatening pneumonias, sepsis, and disfiguring diseases. Most fungi are effectively controlled by host immunity, and they can reside within an individual for a lifetime. But these same fungi can cause serious diseases in your immunocompromised host. Well, antimicrobial therapy addresses that there are unique metabolic pathways and structures of the fungi but may be toxic and requires lengthy treatments. As with bacteria, extensive use of your antifungal agents, such as what you see in the background, in your hospital setting has resulted in the emergence of yeasts and molds that express intrinsic and acquired resistance to several different classes of antifungal agents. Let's talk briefly about the parasites. Well, it comes from the terminology called parasitism, which is a close relationship between species where one organism, the parasite, lives on or inside another organism, which is we call as the host, causing it some harm and adapted structurally to its way of life. An entomologist, E.O. Wilson, has defined this or characterized parasites as the predators that eat prey in units of less than one. Okay, parasites includes single-celled protozoans such as your agents of malaria, sleeping sickness, and your amoebic dysentery. Animals such as your hookworms, lice, mosquitoes, and vampire bats. Fungi such as your honey fungus, agents of ringworm, plants such as mistletoe, dodder, and broom rapes are best examples. This is an image of your Pleunorchis sinensis, a Chinese liver fluke which is trophically transmitted. Another micrograph of your vector-transmitted protozoan endoparasite Trypanosoma among human red blood cells. This is an example of your life cycle of your Entamoeba histolytica, an anaerobic parasitic protozoan transmitted by the fecal oral route. Parasites are the most complex microbes. Although all parasites are classified as eukaryotic, some are unicellular and others are multicellular. They range in size from the tiny protozoans as small as your 4 to 5 micrometer in diameter, which is the size of some bacteria, to tapeworms as big as them that can measure up to 10 meters in length, such as your arthropods or bugs. Indeed, considering the size of some of these parasites, it is hard to imagine how these organisms can be classified as microbes. Of course, their life cycles are equally complex, with some parasites establishing a permanent relationship with humans and others going through a series of developmental stages in progression of their animal host. Parasitic diseases is diagnosed by symptoms. Okay? It could be also diagnosed with a good patient history taking and detection of your microbe. And there are a lot of helpful hints that are being obtained, especially from the travel and the dietary history of the patient. Because many parasites are unique to different global regions, and there are therapies that exists for some but not for all parasites. And the development of resistance to your antiparasitic agents has complicating or is being complicated because we want to prevent and to treat many infections involving your parasites. Next is immunology. It's difficult again to discuss human microbiology without discussing the innate and immune responses to the microbes. Our innate and immune responses evolve to maintain our normal flora microbiome and to protect us from infection by pathogens. Well, we will learn in this particular section this following image. This is your MRSA. This is ingestion by neutrophils. The yellow entity which is found in your screen is an MRSA, methicillin resistant Staphylococcus aureus, that is being ingested by a neutrophil which is colored in purple. This is coming from your NIH labs and clinics. Well, immunology is a study of your immune responses. That there are physical barriers that prevent invasion by the microbe, and we have innate responses that can recognize molecular patterns on the microbial components and activate local defenses, and specified adaptive immune responses that targets invading microbes for elimination and somehow blocking their toxins. Unfortunately, the immune response is often too late or too slow to prevent or limit the spread of the infection, and the ensuing war between the host's protections and microbial invaders escalates, and even when successful, the inflammatory response that result often contributes to or maybe the cause of the symptoms of your disease. And to improve our body's ability to prevent infection, your immune system can be augmented either through the passive transfer of your antibodies, which is present in immunoglobulin preparations, or through active immunization with the components of the microbes. We call them to us your vaccines. And ultimately, the innate and immune responses are the best prevention and cure for any microbial disease. Another aspect in this lesson is what we call as your diagnostic microbiology. Many diagnostic tests require viable samples, and the quality of the results depends on the quality of your specimen that is being collected from your patient. Okay? And this by means is being transported. Okay, this is being transported from the patient to the laboratory, and the techniques used to demonstrate this microbe in the sample is being identified. Okay. In addition, the collected specimen may or must be a representative of the site of infection, and it is being not contaminated during the collection in other organisms that colonize your skin and your mucosal surfaces. Again, antimicrobial susceptibility determinations require viable and representative microbes that is being purified from the clinical sample, and knowing the minimal inhibitory or the biocidal concentrations for specific drugs is really important in prescribing the best treatment. In your clinical microbiology, it plays an important role in the diagnosis and control of your infectious diseases. There are newer molecular, immunologic, and proteomic technologies that are being used to enhance this information that the laboratory can provide. And the procedures for your genome and antigen analysis have become less expensive and available not just for single group but a lot of pathogens. Okay? These procedures may not require viable samples, and these assays are very sensitive and specific, and it can speed up your analysis. Let's talk about microbiology and immunology in the clinic. Some diseases arise when a person is being exposed, again, to organisms from your external sources, and these are called exogenous infections. These exogenous infections include influenza virus, Clostridium tetani, Neisseria gonorrhoeae, coccidioides immitis, and your Entabiba histolica. You can see in the background a photo of a plate of your coccidioides immitis. Most of your human diseases are produced by organisms from the person's own microbial flora that spread to normally sterile sites, for instance, your blood, brain, your lungs, peritoneal cavity, in which disease can ensue, and refer to them as your endogenous infections. Relatively few organisms are classified as always pathogenic. For instance, rabies, virus, Bacillus anthracis, Shigella, Sporotrynx. Some establish disease only in a well-defined circumstance or under certain conditions, such as for the patients who were immunocompromised. These are called opportunistic infections. Some infections can cause a single well-defined disease, which is sometimes caused by the action of your virulence factor, such as your toxins that is being produced by your Clostridium tetani, the one that causes tetanus. Whereas others can cause several manifestations of a disease, such as your Staphylococcus aureus that encompasses all the systems, like your endocarditis, pneumonia, wound infections, and also food poisoning. The same disease can be caused by different microbes, such as your meningitis, that can be caused by viruses, bacteria, fungi, and your parasites. So we will be able to understand the characteristics of the microbe and the host response to the infection. And there is what we call as your Sherlock Holmes-like approach. Okay, if you're familiar with him, it could also be applied. This approach can also be applied to the microbial villain that to solve the clinical infections disease cases. In addition, there are a lot of proper precautionary measures that can be taken to protect yourselves, and a sensible approach to prescribing appropriate therapy that can be designed. There are 4 questions regarding an infectious disease patient in this box, and these must be answered. First, is it an infection? Where is the infection? Which microbe is causing the infection? And how is it causing the disease? Should it be treated, and if so, what is the best treatment? We will discuss more of this in the future slides. The first question and the first step in treating an infectious disease is to recognize and distinguish an infection from other maladies or diseases. Infections are often accompanied by these particular symptoms, such as fever, inflammation, swollen lymph nodes, and other symptoms. You'll see in the table are the indications of an infection. The next question is, where is the infection? And knowing the site of the infection can provide clues as to the possible microbes causing the infection, and is important in picking the antimicrobial that can reach the infected tissue or the site. Next is question number 3. And these are the main subjects of the medical microbiology. Which microbe is causing the infection, and how is it causing the disease? Although the distinction between the bacterial, viral, fungal, and parasitic infections can oftentimes be made from the history and physical presentations of the patient. Certain lab tests can help focus diagnosis. For example, bacterial infections are often accompanied by increases in serum levels of your C-reactive protein and procalcitonin. And these are components of an inflammatory response. And once a differential diagnosis, you can have a list of the most probable villains is being obtained, then confirmatory tests can identify the disease-causing microbe. And there are sections for the medical microbiology that will introduce you the different types of tests and their applications to each of the microbes that is being discussed. And in addition to knowing the most appropriate test for a microbe or a microbial syndrome, it is also important to know the limitations, sensitivity, and specificity of the tests. And the fourth question should take the considerable thought. Should the microbe be treated? And if so, what is the best treatment? Now, designing an appropriate therapy is necessary to those infections that do not resolve on their own. Although safe antibiotic treatments can disrupt the normal flora, it will also allow more pathogenic bacteria and fungi to take place. And proper therapy requires getting enough of the right antimicrobial drug to a sensitive target within the microbe at the site of the infection of your body. And that is what we call as your antimicrobial stewardship. Okay? Initial antimicrobial choices may attempt to cover all the possible pathogens, but on identification of the microbe and its antimicrobial susceptibilities, antibiotics now are more specific, less expensive, easier to administer, and they have fewer side effects, and they should be prescribed. And proper antimicrobial stewardship will reduce the cost, side effects, and potential development of resistant strains. In addition to the four questions relating to the patient, the care provider must also know how to protect themselves, okay, and other people from infection. Key questions include: is there a vaccine? What safety precautions should be taken? How can your hands, your objects, and the contaminated surface be disinfected? And the best means to protect an individual from an infection is to prevent what? Exposure and contact. And the second best means is to be immunized against the microbe by prior infection or vaccines. Also, knowing the epidemiologic characteristics of the microbe will help us to determine the potential for exposure and identify who is at risk of the infection. This includes the means of spread, such as the vector, if it's being utilized in a certain geographical distribution, and the seasonal presence of your microbe, and as well as the influence of your personal health, genetics, habits, lifestyle, which also increases the risk of infection and disease. Asking a patient whether they have traveled recently has become a key question in obtaining the diagnosis as well as the indication of the globalization of your disease. Again, it's really important to realize that our knowledge of this microbial world is evolving continually. Just as the early microbiologists built their discoveries on the foundations established by their predecessors, present and future generations will continue to discover new microbes, new diseases, illnesses, and new therapies. And the following lessons in your medical microbiology intended for us to involve and to form a foundation of knowledge that can be used to our understanding of the microbes and their diseases. Thank you so much for listening. Up next, Human Microbiome in Health and Disease. This is Sir Steven, and have a nice day. Thank you.